Okay, so welcome back friends, welcome back in another video lecture from microbiology with Charnik. In this video tutorial we will going to discuss about gene therapy method. Okay, so in that video we will going to discuss about the two approaches, although one we have already discussed, that was ex vivo gene therapy. So today's goal is about understanding the second topic, that is transfer of gene directly to the cell inside the body, that is in vivo gene therapy. Okay, although the in vivo gene therapy is consists of several topics, so here I will just complete the basic principle and the difference between ex vivo in vivo gene therapy and the target cell for gene transfer. Then the various kind of vector that we are using for in vivo gene therapy, I will going to upload this in the gene delivery video lecture series, and I would like to recommend you to go and watch those video lecture playlist, then it will be good for you. Okay, so now let's start the in vivo gene therapy. So we have completed our lecture about ex vivo gene therapy by discussing about a thing, that ex vivo gene therapy is only possible for those tissues where isolation of the cell is necessary and important and easy, most importantly, and then after isolation, culturing of that cell in the media, and then after culturing and the transfer of the therapeutic gene, the reintroduction of those cell into that tissue back is easy. We are using ex vivo gene therapy only for those kind of tissues, for example hematopoietic cell and skin cells, right? So but gene therapy are also important for various kind of tissue which are present in very delicate part of our body, from where isolation of cell is not possible, and if we can somehow able to isolate them, then culturing of them in culture media, we will not get sufficient amount of cell. And not only that, again another thing, that if we can somehow get a sufficient number of cell, then reintroduction of those cell back into that delicate part of the tissue will not be easy and not also efficient, right? So under such circumstances, in vivo gene therapy works well. Okay, so in vivo gene therapy, as its name suggests, we are transferring the cloned gene directly to the tissue inside the body. Okay, so first thing is that we are transferring therapeutic genes, which is nothing but DNA or a nucleotide sequence, right? So we are transferring it with the help of vector. So as here we are dealing with inside environment of the body, now a appropriate gene delivery method is very important, right? Because in case of ex vivo gene therapy, as we are doing this totally inside in a culture medium, we don't have to bother about the gene delivery method too much, because we can choose any kind of gene delivery method for transferring the transgene or the therapeutic gene. Okay, but in case of in vivo gene therapy, as here we are concerning with the transfer of the therapeutic gene in the inner part of the tissue, it is very important to talk about the gene delivery method. So here we will going to use two kind of gene delivery method. We have only two choices here. One is viral vector, another one is liposomal vector. Okay, these two viral vectors can be useful for transferring those therapeutic genes directly. Okay, now if you are using viral vector, then it is necessary to use a special type of cultured cell known as vector producing cell. Remember, those type of cells are already infected, or that already has been infected with a special type of recombinant retrovirus. Okay, that will give that kind of cell a ability to produce modified virus regularly. Okay, modified viral vector regularly. So this cultured cell will be called as vector producing cell, and that's why the name vector producing cell. So this vector producing cell will allow them to transfer the gene surrounding the disease cell. Okay, so now the efficiency of the gene transfer and the expression of the gene here not 100% possible, because remember it, we are using a particular type of culture cell to transfer them inside the body. Okay, so there can be a chance that our disease cell have not taken up the gene, okay, that is the therapeutic gene. There can be a chance, but in case of ex vivo we don't have to bother about this, because in case of ex vivo gene therapy we are only selecting those cells that have acquired the transgene, right? But in case of in vivo gene therapy we don't have that opportunity, right? So under such cases we have to bothered about the efficiency of the process. That means if the gene transfer method is efficient and if the expression of the transgene inside the cell is working properly, then under such circumstances only our entire work will be considered as successful, otherwise not. Okay, so now we will going to discuss about a difference between ex vivo and in vivo gene therapy. So ex vivo gene therapy will confirm more innovation rather than in vivo. Okay, ex vivo although technically complex than the in vivo type, and vectors are not introduced directly in case of ex vivo, right? We are correcting the cell, then we are transferring the cell, not the vector, but here we are inserting the vector. Okay, now safety check is possible in case of ex vivo, right? Because we have the control to transfer only our corrected cell, right? But here safety check is not possible. Okay, close control possible, decrease control over target cell. Okay, so now as here we are talking about target cell, right? So now it is necessary to understand during doing the in vivo gene therapy on the basis of for curing a particular type of disease, what target cell we should choose. It is necessary to know. So first of all, cancer cell. If you are suffering with cancer cell, then what if you are suffering with cancer, then for destroying those kind of cancer cell either by our prodrug method or by toxin method, what kind of target cell we should choose. So first of all, as they are tumor cells, so tumor cell obviously, then APC, blood progenitor cell, okay, along with T cell. For inherited monogenic disease we will target macrophages, T cells and blood progenitor cells. For infectious disease we will target T cell, blood progenitor cells and APC. And for cardiovascular disease we will target endothelial cell and muscle cells. And for rheumatoid arthritis disease, synovial lining cells are important to target with the help of specific type of viral vector or liposomal vector. And for if we are using viral vector, then along with that a vector producing cell, okay, as a culture cell. So that was the concept of in vivo gene therapy. So remember that in vivo gene therapy is only useful for transferring the transgene into those cells where culturing of those cells in a culture medium is not easy and we will not get sufficient number, and also after that reintroduction of those cultured corrected cell inside the patient cell back will be not easy. That is why here we are using a process that will transfer the transgene directly to the tissue inside the cell. Okay, so for that the gene delivery that we are using are liposomal mediated gene delivery and other viral vectors that we have used. Okay, so now it is necessary to understand about what are the liposomal mediated gene delivery and what are the other gene delivery method present. So for that purpose I would like to recommend you to go and watch my gene delivery lectures video, then it will be easy to you for understanding. Okay, so till then friends, bye bye. So I hope this gene therapy lecture video series will help you to understand about the basic concept of gene therapy. Okay.